First of all, we're going to hear from Professor Joyce Tait, who is Scientific Advisor of the Innogen Centre, or to give that its full proper title, the ESRC Centre for Social and Economic Research on Innovation in Genomics at Edinburgh University. You can see why they say Innogen Centre. She's one of the kind of rare breed, a very broad spectrum, across the board, jack of all trades academics, able to look and see the bigger picture of things, and very good at putting scientific developments in their broader context. There's a lot of work across a wide range of areas, including GM and stem cells, but has recently published Risk Governance of Synthetic Biology. So this is one of the hotter topics right now. We can expect to hear more from her on risks specifically later, but for now I just want to see if she's got any specific thoughts on what you've just heard, Joyce.
No, I think so. I was interested in Jim's point that we've got the capacity to build but not to design. I think we're getting pretty close, hearing what Ben said, to actually designing stuff.
No, I think to build the capacity to build, say for example, from the point of view of construction of DNA molecules that might be used to reprogram cells, we can build a genome right now. If you look at the numbers of genes that are used in currently published constructions, the maximum is in the order of 10 genes, whereas 500 is in the smallest genome. Yeah. So we clearly have capacity that's not being filled by ability to design these things. And there's clearly, if you want to make large-scale changes, that would be potentially useful.
Any other thoughts? No, that's fine, thank you.
Oh, that's right. Okay, well in that case we'll go very swiftly to our final member of our panel, Richard Holloway. For 14 years, Bishop of Edinburgh, but since 2000 he's gone his own way and now is occasionally inclined to describe himself as post-religionist, which itself sounds like a new kind of life form. Among other things, he's a writer and broadcaster and former chair of the Ethics Committee of the Human Fertilisation and Embryology Authority. Richard, your thoughts again.
Right. Well, I don't want to rain on this colourful and glorious parade, but I do want to suggest the importance of a mild April shower on it. The human animal is the cleverest animal on the planet. We've got these enormous brains on this table, excluding mine. But we also have a record of misusing the discoveries that we make. Science's original sin was Hiroshima and Nagasaki. So science can be put to evil uses. What I think we need, therefore, to do is, alongside all this glorious science and the wonderful inventions, the approaches to tackling diseases, plant biology, I think we also need to think about the ethics of what we're doing. And just because we can do a thing doesn't necessarily mean that we need to do it. I think the other thing I'd like to say is that we have this capacity for misusing good things. Think of the muck that's on the internet, which is a wonderful invention. Think of the possibilities of these technologies to do bad things with them. Not to stop it, but I think we should be neither pessimistic nor optimistic. I think we should be realistic that if this is the great new frontier, and I'm sure it is, of science, and remembering what we've done with other great frontiers in the past, we should be cautious, not prohibit any of this, but monitor it, be aware of the misuses that this could be put to, bioweapons, there could be terroristic uses. I know that scientists are very, very, very well aware of all of this, but one of the things that I've observed in the history of science, there's frequently what I call the ethics lag, that you're able to do a thing before you've thought about whether you want to do it, whether you want to limit the doing of it. So all I'm doing at this moment is saying, yes, this is glorious, but let's be careful that it's not put to bad uses and that there are going to be unintended consequences. Let's try and anticipate them. So I hope that our scientists, and I know they are actually aware of all this. They're doing science in an ethical arena and not simply as a pure activity.
You're not pessimistic or optimistic, but you don't also think it shouldn't be over-regulated or under-regulated. We've got to get the balance right.
I think a good example of the way we've done it in Britain is the Human Fertilisation and Embryology Act, which neither let it rip nor did it ban it. It regulated it, and it set up an authority. It permitted experiments in embryos. There was people in the country didn't want that. There are big religious anxieties about all of this, and not just religious anxieties. You're dealing with the building blocks of life. There are people talking about inventing new species, and I'd like to hear from Ben what that might what that could possibly mean, that kind of thing. I think we just need to call canny here and be careful, knowing our capacity to do bad things with good things.
Joyce, you've got something else you want to say now?
I did want to make some points about regulation, following on from that point, because it's certainly the case that this technology will be regulated, and fairly heavily regulated. And if we want to see the full benefits of the technology, we'll actually need to have a much smarter approach to regulation than we've had for earlier types of tech.
Because traditionally there is this sort of, you've talked about the ethics lag, but there's also a kind of legislative lag often, that you're kind of regulating to play catch-up all the time.
Yes, and we've, with earlier versions of GM technology, for example, we've not been very clever the way we've regulated it and the way we've dealt with people's concerns about it. And I think we're actually going to be much better this time round doing it with synthetic biology. But the regulators themselves need to be very aware that the early choices they make about how to regulate new technology can have a very inhibiting effect on new developments that would end up being very publicly useful, but don't get developed because of particular regulatory choices that are made before we really know enough about the technology to know what its faults and defects are going to be.
But just to balance that, as well as a kind of risk of an ethics lag and a legislation lag, do we not also need to be worried about, I don't know what the opposite of a lag is, a gal? I don't know, but it's a sprint, but where we actually get too far ahead of ourselves, like has happened with nanotechnology. We get frightened ourselves about grey goo and demon robots and great creations, and so it's going, hang on, we're just trying to get a couple of molecules to talk to each other at this point, and we're getting our fears way ahead of where we are.
And once it gets into the newspapers, because they're wonderful at stoking up these moods of anxiety, and you can trap yourself in, I mean, look at the recent stupid debate we've had in the country about this latest recreational drug. And of course, don't leave out the political element, because politicians run these things ultimately, and they're very rarely interested in the ethics of the science. They're interested in the electability of a particular process. And hence they tend to jump, I think, too quickly to legislation, usually unintelligent legislation. So we have to educate politicians, which is an enormous task.
Yeah, let's not. Synthetic life, yes. Educating politicians. Let's not get ahead of ourselves here. Ben, you look like you wanted to chip in.
Yeah, well, I think Richard brought up a wonderful example of how things can become dangerous, and perhaps the Manhattan Project was a real illustration of how things can go wrong, and perhaps the atmosphere, the history of the time. The lack of engagement with society, the lack of discussion, the lack of, therefore, of any sort of ethical and moral framework led to people getting to a point where things were wrong. I mean, in my mind, and this is perhaps unfair because I wasn't there, I wasn't in their shoes, but the point at which you become a scientist in a locked compound in the middle of the desert, before you've got there, it must be clear that something is going on. The point at which you are in a project that is entirely funded by military goals. Of course, there were the other moral and ethical considerations of the war, but there comes a point when you realise that control has gone down a very narrow channel. And I hope that scientific discoveries in the context of society, in the context of discussion, the more discussion there is, more often there is a lack of a lag or a sprint, because people can say, we just don't think that's right. And I think most of the scientists I would know are excited about what might happen. They're curious, they can be kid-like, but at the same time, when they've got enough people around them to say, but do we really want to do that? They will be informed by discussion. By definition, the good scientists are open-minded, not just to results, but to the debate around results.